Good morning, students. Welcome back to my channel, Molecular Mysteries. So, I told you in the last class we have covered the important topics regarding the cancer and all. I thought of posting this class genetic code in the transcription and translation itself in molecular biology. I'm so sorry I'm posting this here. Please get to know more about the molecular biology in the classes that I have already posted in the video section. Please go through it, get an idea about transcription and translation, then only you'll get an idea about what is genetic code. Okay, in this class let's talk about what is genetic code and what are the properties of genetic code and how the tRNA really helps in getting an anticodon for the mRNA. Okay, in this class let's talk about what are the genetic codes, what are the types of genetic codes, and how many genetic codes are required or how many codons are required for reading an mRNA for successful protein synthesis and release of amino acids. Okay, so this is all about the protein synthesis continuation. So please go, I'm also linking the main important transcription and translation in eukaryotes and prokaryotes in the description box. Please go through it. Okay, so now let's see what's happening in genetic code. What is a genetic code? So before you get to know it, you should be knowing that genetic code is a 3-number nucleotide sequence which is present on the mRNA, based on which the tRNA would get the anticodon, and finally the release of amino acids will be done. So you all know that there are 20 different amino acids, right? There are mainly 20 different amino acids, and each and every amino acid has got a particular genetic code through which it gets translated. Okay, so now let's see what are the properties also in genetic code and what are the properties of amino acids and how the genetic code would code them and release them. Okay, so hope you're all aware of the central dogma of molecular biology, which is called the transcription and translation combined, right? So that is what you call the central dogma. Okay, so how the transcription happens, based on which the mRNA acts as template, based on which the whole translation happens, right? That is what you call the central dogma. The information that is present on the DNA gets transferred to mRNA and finally into proteins. And here you have seen that it's a rough amount of genes that are present in a DNA. So humans generally have 30,000 genes in a DNA, and the Escherichia coli generally have 2,400. This is just a rough number for your understanding. Okay, now let's see what is a genetic code. So, as I told you, it is a system of nucleotide sequence of mRNA that determines the sequence of amino acids in the proteins. You all know that the building blocks of proteins are the amino acids, right? All the proteins are made up of amino acids. So, these amino acids are generally formed in the cell through the translation process, where the tRNA gets the anticodon sequence from the mRNA. Okay, it's like a dictionary. You got a bunch of genetic codons in your cell through which the amino acid sequence or the tRNA sequence happens or the translation process happens. So genetic code, it is a system of nucleotide sequence of mRNA that determines the sequence of the amino acids in the proteins. It is like a dictionary. It acts as a correspondence between the nucleotide sequence and the amino acids. Hope you got what is genetic code. Okay, let's see how the genetic codes look like. So this is the exact picture and that I have taken from my book, which I really follow. So if you go through the amino acids, as I told you, they have 20 different amino acids. It could be phenylalanine, leucine, serine, proline, right? You have lysine, you have glycine, you have histidine, thyrin, you have arginine, right? These are all the different type of 20 different type of amino acids, and the number that you are seeing here, or or the sequence that you are seeing, is what you call the genetic code. UUUUUC. So uracil and cytosine. So these are all the combinations of the amino acids. That is how the genetic code is been maintained. Okay. So when you see these amino acid sequences on mRNA, the tRNA get those anticodon sequences, and hence that amino acid is finally formed in your cell. Okay. This is how the list looks like. Okay. Now let's see what are actually the codons. So how many codons are there, and how many amino acids have been done by these codons. Let's see into it. Okay, so I told you the codons are the representation of the adenine, guanine, cytosine and uracil. I told you already in the molecular biology itself there are four different types of nucleotides. Based on the arrangement of these, whether it's a triple bond, it's a double bond, based on it the whole DNA is made, right? So based on these codons only you will get the amino acids. From based on the codon sequences you will get the amino acid sequence released in each cell. Okay, so you'll also know you have the 5 prime end and the 3 prime end, and the anticodon having the 3 prime end and 5 prime end. So if you have any questions, please let me know in the comment section. Anyway, I'm adding the transcription and translation process as well in the description box. So when I told you we have 4 different nucleotides, each codon is made up of 3 different nucleotides, right? So nucleotide bases. So based on which we can say that it's a triplet, right? The codon is actually a triplet. So when you have 4 nucleotide bits, the estimation would be 64 codons, right? 4 to the power of 3, and you have mainly 64 codons which are coding for 20 amino acids. But out of which I can say 3 are the nonsense codons where the whole process of translation gets stopped, right? So when the ribosome moves towards the mRNA, towards the end of your mRNA, the tRNA gets to know that it is a nonsense codon from for which it doesn't have any anticodon, right? So it says the ribosome would immediately stop translating the protein, and the process of translation is done when you say they have nonsense codons. So what are those nonsense codons? They are UAA, UAG and UGA. Okay, these could be asked in some of the questions, some of exams that I'm telling you. So UAA, UAG and UGA, these are called the nonsense codons, which would immediately tell or immediately make the ribosome to stop the process of translation. Okay, that is how they are called nonsense codons or the terminator codons. So I can say UAA codes for amber, UAG codes for ochre, and UGA codes for opal. So this is how the non-stop codons would immediately make the ribosome stop and the translation process is stopped. So if you see that out of 64 codons, you got only 61 codons coding for 20 amino acids. The remaining 3 are the nonsense codons. Okay, and now let's get into what are the properties of the genetic code and how the code is really written, like how are they overlapping or they having any commas. Now let's get into know about more about it. Okay, so I told you this genetic code is very specific to each and every amino acid. Now let's see what are the characteristics of genetic code. Specific codon always code for a specific amino acid. I told you they are very specific in nature. When they have a UUU, that would immediately code for phenylalanine amino acid. If you see UGG, it would only code for the tryptophan. So that is how the specificity is maintained in the genetic code. And I told you it's a universality. The genetic codons for amino acids are always the same with a few exceptions of mitochondrial mRNA. So this genetic code for amino acids are always the same wherever it takes. But you have mitochondrial extra chromosomal DNA which is called mRNA even in your mitochondria, right? So that is a little different from normal, but genetic code is generally universal, right? Genetic code codes for amino acid are same with little few exceptions. Now let's see the important properties of genetic code. So I told you these are the non-overlapping codons. So here you can see AUG, CUA, it's continuous, like the genetic code is written like continuous, but it is not overlapping here, right? AUG, it codes for one amino acid. CUA, it codes for one amino acid. So you already know UUU codes for phenylalanine, right? Right. So this is how this is AUG is a start codon for both eukaryotes and the prokaryotes. I can say AUG is a starting codon. Okay, so here you can see there are no overlapping sessions happening. So you have a bar happening in the middle which is coding for particular amino acid. So there is nothing like AUG and UGC, GCU. This is nothing. This is not the overlapping. So how this is how the genetic code is maintained without any overlapping. If overlapping happens, it would lead to some other amino acid coding for some other codon, so it would get disturbed and this may result in very different diseases also. Okay, now let's see how the codons are commaless. I told you codons will always be 3 bases. Always the codons read continuously as 3 bases without any punctuation. Okay, that is how they are called the commaless, and they are non-overlapping in nature. Okay, now let's see how they are called the degeneracy. The coding for some amino acids by more than one codon. So, for example, if you talk about methyl, sorry, if you talk about tryptophan, okay, if in the rest of the amino acids you have only 2, 2, 3, 4, 5, 6 triplet codons only. Okay, the degeneracy is a main important nature of the genetic code, where the same amino acid is coded by more than one codon. Okay, these degenerated codons differ mostly on the third nucleotide. So I told you the genetic code is generally having three nucleotides, right? So the last one would be the deciding factor which amino acid it has to code for, right? So that is how it is called the degeneracy codon, different mainly in the third nucleotide. No significance. Now reduce the harmful effects of mutation. So wobble based pairing. So this is the important wobble based pairing. This pairing would help you to reduce the mutations while having a genetic code reading. Like during the translation process, instead of having any mutations leading to many diseases, this wobble based pairing would really help the genetic code to go in a normal consistent way. Where what is wobble based pairing? So you all know that 61 codons has to really code for 20 amino acids, but it is not like that. The Vobel base pairing is the third nucleotide base in a one codon would be a little stringent. The first two would be very particular. They have to code for the same amino acid, same thing. The third one would be pairing would be based on the stringent thing. Like it could be anything. If G comes in, it would lead to one amino acid. If C comes in, it leads to one more amino acid. Okay. See, this is what you call the Vobel base pairing, which is actually it was prepared by Watson and Crick. You all well aware of the structure of DNA, right? So this is how the wobble based pairing happens. I told you wobble based hypothesis is the mechanism by which the tRNA can recognize more than one codon for a specific amino acid. You got, I hope you got the point. tRNA gets that particular mechanism where it has got you can recognize actually one code more than one codon for the particular amino acid. So codon and anticodon base pairing, that is mRNA and the tRNA base pairing, follows the Watson and Crick rules for the first two bases of the codons, right? So you got to know the point. This is all about the third nucleotide I'm talking about. The first two bases of a codon is always the same, but it is all about the third one, which is a little stringent. Okay, so I can say, but it's less stringent from the last base. So 61 tRNA may not be required to read 61 codons. So it doesn't mean like that. tRNA can get with the less amount of tRNAs itself, we can code for 61 codons, because it's about wobble hypothesis. If you have any doubt, please let me know in the comment section. If you want to have a clear idea, I will also post what is wobble hypothesis in the description. In the next class, I will talk about the immunology, because till now we have covered the cancer topic, oncology topic, and all basics on. In the next topic, let's talk about the immunology. Immunology is also important, where we have to discuss about the immunity, types of cells, where are the cells produced, okay, and how the mechanism happens. Okay, these are all where are the cells produced in bone marrow, and what's the mode of action of T cells and B cells. Okay, in the next class, let's discuss about it. So hope you like the class. Please let me know in the comment box section. Thank you and have a nice day.